I'd like to thank the sponsors of this really terrific meeting for the opportunity to be here today and tell you a little bit about Iconic. We're targeting the tissue factor pathway to address high unmet medical needs in both ocular disease and oncology. I'll be speaking today about our lead program called ICON-1, which is advancing in clinical development for wet age-related macular degeneration. We are also quite excited about our oncology program with our second molecule, ICON-2, which is entering IND-enabling studies over the next 18 months. We're doing this work with a select team of industry veterans who have all been inventors or developers of first-in-class biologics, including in ophthalmology, and we're quite grateful for the strong support from our investors. As we all know, wet age-related macular degeneration is a devastating disease, and unfortunately, for a significant portion of patients, current therapies do not prevent continued progression to end-stage visual loss and even blindness. In particular, anti-VEGF treatment, while quite effective in reducing exudation from choroidal neovascularization, cannot address the underlying process of the production of angiogenic factors, nor the inflammatory component of the disease, and benefits are only accrued with continued and often frequent injections. We firmly believe that a more fundamental treatment paradigm is required that could halt disease progression and delay or prevent long-term visual loss. Tissue factor is a normal human protein that's overexpressed in several disease states. In terms of macular degeneration, tissue factor is expressed on the CNV blood vessels themselves and in the vitreous and retina of AMD patients, and not in normal eyes. Tissue factor expression drives both inflammation and angiogenesis, and there's a positive feedback loop between tissue factor and VEGF. Thus, inhibiting tissue factor could offer a more comprehensive and potentially more permanent solution. And our lead molecule, ICON-1, targets tissue factor to hopefully do just that. So how does it work? We know that anti-VEGF agents are quite effective at eliminating soluble VEGF present in the vitreous in AMD patients. ICON-1 binds to tissue factor on the CNV itself, therefore potentially inhibiting release of VEGF and other angiogenic factors, reducing inflammation, and may reduce or remove the CNV lesion itself. Thus, together, we have the opportunity to reverse both the temporary visual acuity losses due to exudation and reduce the drive to permanent vision loss due to persistent CNV activity. The recently completed EMERGE Phase IIa trial demonstrated this potential. EMERGE was a randomized, double-masked, active controlled trial conducted in the United States in 88 newly diagnosed treatment-naive wet AMD patients. Patients were randomized to one of three treatment assignments. 1, ICON-1 in combination with Lucentis, 2, ICON-1 alone, and 3, Lucentis as an active concurrent control. The study was divided into two portions. In the first induction phase, patients received their randomly assigned treatment every month for 3 months of induction therapy. In the second 4-month follow-up phase, patients remained in their assignment treatment groups, and treatment was given by physicians based on their clinical assessment of continued CNV activity and disease symptoms, thus affording us the opportunity to look both at anatomic and clinical outcomes, as well as the durability of the induction therapy phase. ICON-1 was well tolerated. There were no serious ocular adverse events, and other safety outcomes were as expected in any trial of intravitreal therapeutics in this population. Turning to the activity outcomes, I'll first note that ICON-1 monotherapy at the dose given in this trial, while safe, had outcomes on the efficacy side not quite as robust as VEGF alone, as anti-VEGF alone, and we'll be focusing on combination therapy. The key outcomes are shown on this slide. First on the right, visual acuity, not unexpectedly increased in both the Lucentis group and the combination group to about the same degree over the course of this study. The addition of ICON-1 produced several outcomes on disease activity, however, including reduced CNV activity, as demonstrated on the left by 3 times as many patients achieving complete resolution of all pathologic fluid in and under the retina, and reductions in CNV lesion size. In particular, no patient in the combination group had progression of their CNV lesion over 6 months, in clear distinction to the anti-VEGF control group. Further, these benefits were achieved in a more durable way, with 40% of patients in the combination group requiring no additional therapy during the 4-month observation phase, where the vast majority of Lucentis patients required additional injections. We're quite encouraged by the results of this first multi-dose trial of ICON-1. And in addition, we believe there's more room to grow. Animal studies have demonstrated that there is potential for continued and increased activity at higher doses, and doses up to at least 0.6 milligrams have been shown safe in a pilot human study, so we'll be progressing to higher dose studies in the future. In conclusion, we believe there's a need for a fundamental change to the treatment paradigm in wet AMD that goes beyond the treatment of acute exudation to address the underlying processes driving inflammation and CNV growth, together, which drive disease progression and end-stage vision loss. Tissue factor inhibition with ICON-1 has the potential, in combination with anti-VEGF agents, to prevent both initial exudation and reduce the drive to end-stage vision loss, and we're very excited to advance this treatment, future treatment for patients. Thank you.